Good day, everyone. Today, I will be talking about the vital role of baker's yeast in the baking industry and its broader application. First of all, baker's yeast, mainly Saccharomyces cerevisiae, is essential in baking for leavening bread through fermentation to produce carbon dioxide and ethanol. It naturally occurs on fruit skin and plant surface and is available commercially as a active dry, instant, fresh and liquid yeast. Yeast production involves cultivating strain in bioreactor with nutrient-rich media under control condition. Beyond baking, uh, Saccharomyces cerevisiae is used in brewing ale-type beers and fermenting grape juice for wine, highlighting its broad application and significant impact in food production.
Assalamualaikum warahmatullahi wabarakatuh. Today I would like to explain about Saccharomyces cerevisiae metabolism pathway. Saccharomyces cerevisiae, commonly known as baker yeast, exhibit a complex and well-studied metabolic network. This network includes various pathways for the production of energy and biomass, essential for the yeast growth and survival. Here are some key of the key metabolic pathway in Saccharomyces cerevisiae. This is the process by which glucose is converted into pyruvate. During the glycolysis, a molecule of glucose is broken down through a series of enzymatic reactions to produce pyruvate, ATP, and NADH. The pathway occurs in the cytoplasm and is crucial for cellular energy production. In the absence of oxygen, Saccharomyces cerevisiae undergoes fermentation. Pyruvate, the end product of glycolysis, is converted into ethanol and carbon dioxide. This process regenerates NAD plus from NADH, allowing glycolysis to continue producing ATP anaerobically. During aerobatic condition, a pyruvate is transport into mitochondria and convert into acetyl-CoA. Acetyl-CoA then enter the TCA cycle, also known as a Krebs cycle, where it is further oxidized to produce NADH, FADH2, and GTP. This cycle is central to cellular respiration and energy production. The NADH and FADH2 produced in the TCA cycle donate electron to the ETC, which is located in the inner mitochondrial membrane. The flow of electron through the ETC lead to the production of a large amount of ATP through oxidative phosphorylation, utilizing oxygen as the final electron acceptor. This pathway operates parallel to glycolysis and serves two primary functions: the generation of NADPH for biosynthesis reaction and the production of ribose 5-phosphate for nucleotide synthesis. NADPH is essential for reductive biosynthetic process and for maintaining cellular redox balance. This is the synthesis of glucose from non-carbohydrate precursors such as lactate, glycerol, and amino acid. Gluconeogenesis is particularly important during periods of low glucose availability, allowing the yeast to produce glucose for vital cellular function. Acetyl-CoA serve as a building block for fatty acid synthesis. Fatty acids are essential component of cell membrane and are also stored as energy reserve. This process involves the breakdown of fatty acid into acetyl-CoA unit, which can then enter the TCA cycle for energy production. Beta oxidation occurs in the mitochondria. Saccharomyces cerevisiae can also synthesize and degrade various amino acids. Amino acids are necessary for protein synthesis and can also serve as intermediates in another metabolic pathway. These pathways are responsible for the synthesis nucleotides, which are the building block of DNA and RNA. Proper nucleotide metabolism is essential for cell growth and division.
Good day everyone. My name is Sivanathan Jagar and I'm from Sydney. Today I want to talk about development of kinetics for the production of baker's yeast, typically the Saccharomyces cerevisiae, which is the most widely used yeast in the production of bakers. So to begin with, let us talk about the yeast growth curve. In our study, we monitored the growth of yeast in the examples of 600 nanometer to time, and we got this plot which includes the lag phase, exponential phase, stationary phase, and that phase. Understanding this curve is crucial as it allows us to optimize conditions for the maximum yield growth. Now let's dive into the calculation part that support our finding. We started with from our journal we actually found out that the experiment is started with the initial biomass concentration of 0.1 gram per liter, which increased to 1.0 gram per liter after 10 hours. Simultaneously, the substrate concentration, which was initially 10 gram per liter, decreased to 5 gram per liter after 10 hours as well. So this is all our findings, which include the specific growth rate, which is the mu max, the learning time, and the biomass yield coefficient. So right here I have my formula written down for mu max, which is the specific growth rate, the mean time, and the biomass yield coefficient. So from our first finding that already stated here, we could actually calculate this as ln xt over x0 is ln the final concentration of the biomass, which is 1.0 over the initial concentration of the biomass, which is 0.1 gram per liter, over the total time, which is 10 hours. With this calculation, we should be able to get our answer to 0.23 max. And then same goes to the doubling time. As we already got our max, we should easily calculate our doubling time, which is ln 2 over max, which is 0.23, and the answer should be 3.01 hour. For the specific yield coefficient, from the data given, it says that the initial biomass is 0.1 and will increase to 1.0 after 10 hours. So the calculation should be 1 minus 0.1 for the biomass concentration over 5 minus 10 for the substrate concentration with a negative in front, and we should get the answer of 0.18 gram per gram. So up next we have the calculation of degree of reaction, where we mainly calculate for reactant and products. So for the key element of the reactant, which is the glucose, has the number for carbon of 4. For hydrogen it has the number of 1, and for oxygen it's negative 2. So we evaluate the equation and we get 24. And to find the gamma, which is the degree of reaction, we should divide the sum of all the elements in the molecule and divide it by the number of carbon. So we get 4 for the degree of reaction. for reactant, which is the glucose. Same thing we did for the product, which is the biomass of yeast, and we got a value of 4.035. And as we all know, as the degree of reaction increases, the oxidation rate decreases. So therefore product which is the biomass has higher degree of oxidation than the reactant, which is the glucose. Right now we can see the elemental balance equation that was actually found from our main journal. This equation shows the growth of Baker's yeast on glucose that requires oxygen and the reactant. The products are yeast biomass represented as C6H10NO3, carbon dioxide and water. The equation actually happens as an anaerobic respiration, where the oxidized glucose with oxygen to produce energy in the ATP form. The metabolic byproducts of this equation, which are water and carbon dioxide, are typically outputs requiring management in industrial processes.
Hello everyone, I'm Siti Nomaisarah, and today I'll be discussing a mini project on industrial biofuels, biochemical and enzyme production with a focus on Baker's yeast. Let's dive into the key points covered in the project. Firstly, the project outlines the process flow for producing Baker's yeast. The initial step involves cultivating a pure culture of yeast. This pure culture is first grown in a flask and then transferred to a pure culture fermenter. The next step is preparing a sterile mash, which involves clarifying and sterilizing beet molasses and mixing it with nutrients. This mash serves as the growing substrate for the yeast. Once the mash is ready, the seed fermentation begins. The seed yeast from the pure culture fermenter is added to the sterile mash and allowed to grow in a seed fermenter. This culture is then further developed in a semi-seed fermenter before moving to the tray fermenter for final development. After development, the yeast is collected and undergoes a washing and separation process to remove impurities and cool the yeast cream. The clean yeast cream is then subjected to a second round of washing and separation. Finally, the yeast cream is formed and filtered, and these yeast solids are separated using rotary vacuum filtration. The resulting yeast cake is extruded and into the desired shape, dried and packaged for distribution. The microorganism used in this process is Saccharomyces cerevisiae, a single-cell microbe commonly used in baking and fermentation. This yeast consumes sugars in the dough, producing carbon dioxide gas that helps the dough rise, resulting in light and fluffy bread. It is also used in the production of beer, wine, and other fermented beverages. During the aerated fermentation of glucose, both extracellular and intracellular amino acids are produced. Intracellular amino acids are created within the yeast cells, while extracellular amino acids are produced outside the cells. Factors such as sodium chloride and ACL levels in the growth medium can influence the production of these amino acids. For instance, higher NaCl levels can enhance the production of specific amino acids like glutamine, citrulline, and ornithine, which are important for yeast metabolism and overall quality. In summary, this project provides a detailed overview of the steps involved in producing high-quality Baker's yeast, the type of microorganism used, and the production of amino acids during fermentation. Thank you for listening.
And now I will be discussing an exciting approach to improving the production process of Baker's yeast, which involves genetic modification to enhance stress tolerance. First of all, improving the production process of Baker's yeast can be achieved through genetic modification to enhance stress tolerance. Large-scale fermentation subject yeast cell to stresses like high temperature, osmotic pressure, and nutrient limitation, reducing their viability, efficiency. By genetically engineering Saccharomyces cerevisiae to overexpress genes related to heat shock proteins or osmoprotectants, yeast can better withstand this condition, maintaining high metabolic activity and cell viability. Enhancing pathway for allose and glycerol production further improve stress resistance, resulting in higher biomass yield and consistent product quality. This optimization reduces downtime and operational costs, allowing for the use of more variable raw material and enhancing the sustainability of yeast production. That's all from me. Thank you so much.